Alright, folks, we're getting started. So, officially get started. Hopefully you've already started talking about this.
Yes, ma'am.
It might help if you think of a nutrition label, because nutrition labels actually have 3 important macromolecules that they keep track of.
Oh, we learned about this. I don't remember.
Yeah, if we look at a nutrition label, what are some of the categories? Sorry? Carbohydrates. Yeah, fats, which are lipids.
Oh.
Yeah, nucleic acids aren't on the nutrition label, but they're the 4th category.
I didn't remember that.
Okay, so those are the macromolecules. Those are I heard somebody use the word polymer, so these are all polymers, which are made up of monomers. So do you remember what monomers make up carbohydrates? Yeah, what's the prefix mono mean? 1. What's the prefix saccharide?
I don't remember.
Sugar. So 1 sugar, a single sugar. You put them together, you get a polymer. Starch, cellulose, glycogen. What about proteins? Lipids don't really have a true monomer, but they are sometimes made up of fatty acids. What about nucleic acids?
Nucleic acid.
So this isn't up here, but I think it's a good thing to go over. When we eat foods with lots of carbohydrates, why do we need carbohydrates? Energy. Yeah. So carbohydrates are short-term energy. When we eat food with a lot of fat, we also get a lot of energy, but it's more long-term. Yeah, long-term energy. Nucleic acids, that includes DNA and RNA. Why is that important?
It helps with the cell division stuff, right?
It does. We copy it before we divide. We just talked about that. But we need a copy in each cell because that DNA serves as our set of instructions. Yeah. Specifically, the instructions to build proteins. Okay. What do proteins do? They help transfer stuff. They help break down carbohydrates. Well, but they break the carbohydrates down to get the energy out of it. Yeah. They're enzymes. Yeah. And so proteins do all kinds of different things. Pretty much everything that we need to get done in the body is done by the protein. So this unit is going to be a lot about DNA. We have to learn about DNA first so that we can understand how we use it to build proteins. So we're actually skipping to chapter 8 in our textbook, but this first section, our first test is going to be really over the DNA, and then the second test is going to be how that DNA is used to make protein. So that's what we're going to be learning about over the next 3 weeks or so. If you haven't grabbed your folder out yet, go ahead and grab your folder out.
Oh, sorry.
Ow.
What's up?
Imagine wearing shorts in the
Imagine wearing shorts in the winter.
Yeah.
Imagine having feelings in your leg.
I think that's a good thing.
That'd be. He said, imagine wearing shorts in the winter. I said, imagine having feelings in your leg.
It's cold
Oh,
outside.
my legs. I got it.
Literally outside, maybe. Yeah.
Every day. One more.
Yeah. Imagine. I'm positive.
Imagine what?
I'm gonna be imagine. Oh, I thought you thought.
Okay, so your next blank page should say chapter 8, part 1, page 1 at the bottom should have this diagram of mice on it.
You're so cute. You
Yeah,
know?
how to jump from like chapter 5 to chapter 8?
I just like keeping it better.
Chapters are never in order. Not a single chapter.
Well,
Except for math.
I just like teaching it this order in bio, but some people don't.
Last semester we went from chapter 4 to 13.
Okay, so here's the deal. We're going through 1, 2, 3 experiments today that were vital in our understanding of how important DNA is and the fact that DNA is what holds the instructions and not any of the other molecules. For science. So, here's the deal. Well, it's better than injecting it with people.
Oh, no, I'd rather have
Injected with people?
We don't
So, here's the deal.
Some people deserve it.
No, nobody deserves that. Okay, so here we go. Let's refocus, please. As has been pointed out, we're using mice in this experiment and 2 strains of bacteria. Okay, so some bacteria cause disease and some bacteria don't. In this case, we have one strain that does. So the word virulent means causes disease. And so when we give that to the mouse, what happens?
Yeah.
Not only that, but if we cut the mouse open, we can find that virulent bacteria, right? So that's what's killing the mice is the bacteria strain. Poor mouse.
That's working on my paper.
Non-virulent means it doesn't cause disease. So when we inject that into our mouse, what happens? So he's fine. There's no bacteria in the mouth. We take those bacteria, the virulent kind, and we heat them up to the point where we kill the bacteria. So when you heat, the reason things die when they get heated up, assuming you're not like burning them to pieces, is that the proteins unfold. Okay, and when the proteins change shape, that also means that they aren't no longer able to do their function, right? Structure is important for function. So when they put that in the mouse is just fine, right? So this bacteria that killed the mouse in the first set of experiments, when we heat it up and kill it, doesn't have any effect. With me so far? So far, I hope this makes sense. We put in a deadly bacteria, it kills the mouse. We put a non-deadly bacteria in, the mouse is fine. We kill the deadly bacteria before we inject it, no issue. Here's where things get weird. We take these bacteria, dead ones, and non-virulent bacteria that are still alive, neither of which kill the mice, right? But when we mix them together and put them in the mouse, what happens? You're right, it doesn't, does it? So what happens to the mouse? It's dead. And what do we find in the mouse? Live virulent bacteria, which we did not put into the mouse. So, hypotheses. Brainstorm with your group. Go. What do you think could happen?
I think the mouse itself is scared of the needle so bad.
Stop. Excuse you. a black marker?
Okay, I'm gonna ask you your group to go around and give you your group's best idea in just a few seconds. So be ready.
Do you have a marker?
Wait, what's the question again?
I just said,
Why do you think that?
because of the heat.
Oh, yeah.
I think it's right now the book is
Something counteracts something.
Because it reacted
Because-
to everything else.
They do it like that. You should stream it.
I put, it could have, you should.
I'm putting my hair even on.
Okay, you ready?
Huh?
Ready? 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 Okay, who wants to go first?
We will.
Okay.
No, we will. Anyway,
Hey, listen,
you're a loser.
I don't want any repeats.
We basically discussed that we think that both viruses counteract each other. So with the heat, it's kind of like since it's he killed it, maybe the heat kind of killed from the virulent, killed the non-virulent. Okay. And then yeah. Okay.
In that corner, you wanna go?
Viruses mutate really easily, so we thought that maybe the non-virulent copied the virulent.
Now, these are technically bacteria and not viruses. So, but a mutation could be a possibility. Yeah.
No.
So something reacted in the living bacteria? Okay.
We're gonna say the same thing.
Okay. Any other ideas? Yeah.
It was essentially the same as their idea, where somehow the non-virulent ended up adopting the properties of people.
So adopting properties. That is, how would it possibly get those things from the other bacteria?
So mutation.
Okay. Yeah, but how would it know to make that mutation? Hey, hold on. Somebody else was talking. Okay. Okay. Yeah.
So maybe when the cells were like redoing, you know what I mean, like they were going through the cell cycle, they like combined, and when they split apart, it's a new cell that like they kind of adopted the virulent action.
Okay. Okay. So I'm going to tell you Griffith's conclusion here that there is some sort of transforming, what he called a transforming factor.
Oh, yeah, you're right.
Basically, some type of molecule was being passed from the dead bacteria to the living. So some type of molecule was passed from the dead to the living bacteria. That therefore gave that non-virulent bacteria now virulent properties, right? So some kind of genetic material was passed from one bacteria to the other, and that had the trait, the information for that deadly trait. So it's not really changing its own material, it's just getting new material from the dead bacteria. So why did he call it a transforming factor? Yeah. It's literally transforming the bacteria from non-virulent to virulent, right? So I'm going to show you on the next slide here. This is something that happens between bacteria all the time. We now call it horizontal gene transfer. And so I have a video of this. This is from just a couple of years ago. I think it's a pretty cool video. Really short, more like an animation. Okay, so these are bacteria that are alive. This is DNA from dead bacteria. You can literally see this little structure coming out and grabbing and pulling it back in.
That's creepy.
So pretty, a little creepy, I think kind of cool.
Yo, what is that?
It's like a bean.
It looks like
Like a tractor beam, like
Yeah,
pulling
like
in.
Jack and the Beanstalk bean.
no, it looks like a snake's head and
A little bit. But, so,
Yeah.
main point though, before we move on to the next
Look
experiment.
at it. It looks like a head, like a body of a snake. It's just it's just the head.
It's just the head.
Okay, so,
Don't ever
transformation happens. So when Griffith did this experiment, we found out that we could pass genetic traits from one type of organism to another. But we still don't know what the molecule is. Okay, best guesses so far is that it's either DNA or protein. In fact, most scientists at the time actually thought it was probably the protein because it's just such a much more cooler molecule. It's made up of a lot of multiple different options, whereas DNA is pretty boring. It's either A, T, C, or G. But those were the options. So when we go into this next experiment here, we're trying to figure out, is it DNA that holds the genetic traits, or is it the protein? Okay, so keep that in your mind as we take a look at the next experiment on the next page. So, before we dig into that diagram, you take a look up at the screen here. Instead of using bacteria and mice, we're now using viruses that infect bacteria. So, the bacteria are now getting the bad end of the stick here. If you don't know very much about viruses, just a little background information. So here's a picture of a virus. You can see it's only made up of, well, it's a little bit simplified, but DNA and protein, and also lipids like a cell membrane. That's why soap works on viruses, because lipids and soap like breaks down lipids. But anyway, genetic material, protein code. The virus we're using in this experiment is called a bacteriophage because it infects bacteria. Here's the life cycle. So this is the way it works. The bacteriophage lands on the bacteria. It injects its genetic material. The cell reads that genetic material like its own and starts to make proteins, makes new virus. Okay, so all the virus parts get made. The virus parts self-assemble. And when the cell gets packed too full of viruses, then the cell breaks apart and releases more virus. And this is pretty much how the life cycle of most viruses. Sometimes they don't do this right away. They can lay dormant for a while. But even COVID is a similar. It's got a protein coat and then it's got genetic material on the center. It's RNA instead of DNA, but it works in a similar way. So questions on that before we look at the experiment? Okay. So this is a kind of a complicated diagram. If you look at the question underneath, the first question underneath says, what were they able to tag with radioactive sulfur? So in batch 1, we're growing the phages, we're growing the viruses in radioactive sulfur. The only macromolecule that contains sulfur is protein. So when we grow the viruses in radioactive sulfur, we're essentially tagging their protein. Okay, so it's pretty easy to measure radioactivity in substances, so we can kind of track the protein. In the second batch, this time, instead of using radioactive sulfur, they're using radioactive phosphorus. Phosphorus is used to make DNA. Nucleotides, if you remember, is a sugar phosphate in a base. We'll talk more about that tomorrow, but DNA has phosphate in it. So when we grow the viruses in radioactive phosphate, we're making its DNA radioactive. So One set of viruses grown with radioactive proteins. The other set is grown to have radioactive DNA. Questions so far? So we grow the viruses so that half of them have radioactive proteins, half of them have radioactive DNA, and then we let them do what they normally do. We put them with some bacteria, they inject their genetic material, so that's what it's saying here. We mix the radioactively labeled viruses phages with the bacteria, so the viruses are going to infect the bacterial cells, they're going to transform them, pass on their genetic material. Number 2, we put them in a blender. That agitates it, so that what's happening here?
They're mixing.
Well, we're separating, really. We're shaking off all the viruses. So we're separating the leftover virus stuff that's outside the bacteria from the bacteria itself. We centrifuge it. Does anybody know what that means? You spin it around really fast, and that separates it out. So we have the liquid with the virus, extra virus parts. From the bacteria, infected bacteria. And this is in color, so it's a little bit easier to see. When we have viruses with radioactive protein, the radioactive part ends up in the extra virus parts. Does any of the viral protein end up in the bacteria? No. So, we know viruses do not pass on their protein. When we do this with the other half of the viruses, the ones with the radioactive DNA, the radioactivity ends up in the bacteria, the infected bacteria, and not in the extra virus parts. So that tells us that what ends up infecting the bacteria, the DNA. So viruses pass DNA into their host cells. It's the DNA that passes on the traits, not the protein. The DNA that causes transformation, not the protein.
Right, so for the third question below the diagram, it says what kind of material, I think it's genetic material.
Yeah, specifically DNA, right? So the point of this is that it's not the protein that gets passed on, it's DNA.
And we know for batch 2.
Yes, because in batch 2, the radioactive DNA was found inside the bacteria. Yep. So, conclusion, viruses infect or pass on genetic info by inserting DNA and not inserting protein. Yeah, like I said, I know that's a pretty complicated process. So any questions on that one? The last one's much simpler and much easier. Okay, part 3. Paragraph 7. isolated a bunch of DNA samples from like multiple different organisms, including humans. And when he looked at the chemical composition of those molecules, he noticed that there's 4 different bases. So this might be familiar, A, T, C, and G. Those are the bases that you can find in nucleotides. He did a chemical analysis of each sample. And so in this particular case, 30.9% of the nucleotides had A, 29.4% had T, 19.9% G, and 19.8% C. So in your groups, do you notice any pattern? What does this data tell you about the ratio of nucleotides?
It's all decreasing. It's all decreasing. All the data is data.
It's all.
It's decreasing from 30 to 19. Yeah, exactly. It is down to 1. I think we need to be down from
top to bottom.
That's how we read it. Yeah, I remember A is she literally told us from A to decreasing its smaller. See how it's
You like
it's less like
Okay. So the amounts of A and T are really close to each other, and the amounts of C and G are really close to each other. So there's a 1-to-1 ratio between A and T, and a 1-to-1 ratio between C and.
How is that right?
You are right.
That's what I said.
No.
I said that both ways.
Now we know this is we know this is because that's the way the bases pair together, and the DNA structure will take a look like that tomorrow. There is a math problem here for you though, so take some time in your group to figure that out. Yeah, over the math.
I don't know. Wait, is that ratio?
Yeah, so really it's like 1A to 1T and 1D to 1C to 1D.
Is it the force?
Huh? Okay, it's also important to note that these are percentages, so they're all going to add up to 100.
Wait, no, do that.
I really get it. I don't know.
Do we need some guidance?
Yeah.
Okay.
Is it like supposed to be like the other one?
Yeah, it's going to be similar, but
28,
so, well, we're going to go a little more. So, A equals T. So if A is 28, then T is also going to be around 28. Okay. The rest of it has to be out of 100%.
Oh, you just add 28.
So 54. So it's like 46 divided by 2.
Well, so this is 56%, which means this has to be 44%. And these, so these have to be 22.
Imagine getting that wrong, Izzy.
At least I said something to
True. Well, I'm in geometry. I'm not.
Yeah,
We both are.
Well, that's okay. So now hopefully you know how to do a problem like that. Questions on that? Okay, let's wrap it up
I had
then.
a test today and I didn't finish it.
Okay. No, well, there's a quiz I want to give you some time to work on.
It's one of those easy ones.
Yeah, it's not bad. It's not bad. Okay. So, let's see if we can fill in these blanks here to summarize the main ideas. Number 1, Griffith discovered a
No, transforming.
transforming factor.
How do you remember?
Transformer?
Because he's smart.
I like
Sounded like
transformers.
one.
I'm not good at remembering stuff.
I can tell.
A molecule that could be transferred and carry genetic info. I.e., cause physical traits, right? Or we don't have to write that down, but
Info.
remember, we took our regular non-dangerous bacteria and we transferred the genes of the deadly bacteria. We transformed it. Number 2, Hershey and Chase used bacteria and bacteriophages, which are viruses that infect bacteria, right?
What is that one said?
This one?
Yeah.
Genetic info.
No, that was.
Oh, viruses. It's a type of bacteriophage. Oops, I spelled it wrong.
I can't see.
You have glasses?
And then
Yeah. It's so.
Katrina, so bacteria, bacterio, and then
Phage.
I'm looking at it at an angle.
Okay. All right. So when they did that experiment, they tagged the DNA, they tagged the protein. What ended up going from the virus to the bacteria? The DNA. So that transforming factor was DNA and not protein. That's the whole point of that one. And lastly, Shargoff isolated different types of DNA nucleotides and came up with the rule that A equals T and
C equals a G. I mean,
C equals G, or G equals C, vice versa.
Are we done with you?
Got the notes? Up with the Chromebook.
No.
You can definitely take it at least once in 10 minutes.
You get away on What dog you want?
It's 8 points.
Oh.
Okie dokie. What is okie dokie?
Okay. Survive.
Oh.
Really?
It was dead. He didn't say okie dokie.
Then that means okay.
Well, I thought it'd be death. It says death and okie dokie. It'd be like, no, the 2 options are death and okie dokie. Something serious and not serious.
Is it like
It would be like sleepy or okie dokie.
Yeah, that'd be fun.
If I was at nighttime, I would hear that.
Have you done flashlight? Have you done flashlight?
And then I think the next one.
I put DNA, but I don't know. I don't know.
Phosphorus. I think phosphorus.
What's phosphorus?
Your face.
I was funny. That's 49. What now?
You see that?
Here's 22. Minus 22.
Get out of here.
Wait. You're very strange. Okay, 49 is 49. I've been doing babies and bears.
Two is not perfect. Because I did it.
Yeah, don't forget you'll get it wrong.
I missed it. I got a 4.5. No, you can take it sponge back.
Yeah, you can take him all the time.
Only you put up.
I got it wrong.
Alright, what was the answer for the last one?
The numbers are random on that last one.
Well, have fun. We're all getting that one wrong, I guess.
I have a question.
Yeah.
Number 8, you have like the percent sign, you have to.
No, no.
It worked for me though.
What I suggest doing is if you look back at that practice problem we did, set it up like that. And you'll get it right.
Unless,
Like this.
no, I'm confused though. So if, because I was doing 28, so I thought it'd be 28 the next number, that would be
Well,
equal to it.
T would be 28. But it asks for C.
Oh.
I'm bad.
You know what? I didn't.
The okie dokie.
That's cheating.
What's the number 8? Okay.
Sorry, I should have just said something else.
No, I didn't.
I got beat.
Like if you did.
That's not creepy whatsoever.
I forgot what number 6
I took it the first time and it was
Watch me take it down.
Candy Danny. It still has it up on it. Which is 29.
It better be right.
I got the one right.
That's your thing.
Yeah, I did. Because I want to get everything right. So you just duplicate the file.
Are you done?
No, I'm done.
She can know what.
No, when you don't feel like acting for time, and it has multiple times, you just do it once, make a random thing, and then do it. It's not excuses, it's just.
I don't know. Now we have 3 minutes.
That's not, no, it's not even funny. I
Based on-
I forgot. I'm having my mouth. I have shorts. Because they were already in their
Bro, my thoughts so good. And I wasn't paying attention.
The questions have different numbers in them.
I found another way of figuring it out. All you have to do is find out whatever number it takes to get it to 50.
Yeah, exactly.
It's protein, right? Like, yeah.
Yes.
I don't know why they expected me to get up.
What's the answer?